All right, thank you very much. So since 2018, the University of Bonn pools expertise in sustainability research across the faculties in the transdisciplinary research area, the TRA, Innovation and Technology for Sustainable Futures. And the TRA also organizes this lecture series, the lecture series on innovation pathways to sustainability as a forum for high profile and internationally visible scientists who are active in academia or at the science policy interface. We ask speakers to address research opportunities to enhance sustainability in all its dimensions, that means social, environmental, and economics. Speakers are invited to present cutting-edge research in their fields of expertise and discuss related future research agendas with the audience. And for today, my colleague Ute Votknecht invited Jill Farrant. I'm handing over to you, Ute, to introduce our today's speakers. Thanks very much.
Thank you, Ute. And it's my pleasure to introduce Jill to you. She is a professor at Cape Town University. She was born in South Africa and she received her PhD in plant science from the University of KwaZulu-Natal in Durban. Already at that time, she was interested in seeds, and so she did a postdoc at the National Seed Storage Laboratory in Fort Collins, USA, after which she went back to South Africa, where she became first senior lecturer and then associated and full professor at the University of Cape Town. She currently holds a prestigious South African research chair as head of the plant stress laboratory at the Molecular and Cell Biology Department at the UCT. She has received multiple honors and awards, and I only would like to name some of them. So, for example, the Harry Oppenheimer Memorial Trust Fellowship, which is considered the highest research award of Africa, the Erma Hamburger Laureate from the EPFL Wish Foundation, or the L'Oréal UNESCO Laureate for Women in Science. And indeed, since 2014, she is now jury president of the L'Oréal UNESCO for Women in Science program for Sub-Sahara and South Africa, respectively. She is an internationally acknowledged expert in the field of plant desiccation tolerance, and we will hear more about it from her talk. And it was in recognition of her outstanding academic records in this field that last year she received the Georg Forster Research Award from the Alexander von Humboldt Society. And within the framework of this award, her group and mine want to closer investigate the role of plant hormones in early drought response and vegetative desiccation tolerance. And the ultimate goal that is driving the research and is generally also driving Jill's research is to improve crop breeding by the knowledge gained in these studies. Due to these Georg Forster Research Award, she will be a recurring guest here with us in Bonn. So she will come again and again in the next couple of years, and which means that you all should have a chance to also meet her in person in the future. And we are now starting off with this lecture, which she is giving from her hometown in Cape Town. And with this, I would like to hand over to Jill, and we are looking forward to hear from you about vegetative desiccation tolerance.
Thank you so much, Ute, for that really kind introduction. And I'm feeling very privileged to be able to talk to you all today. I hope I do it justice. I'm going to stop my video and leap straight into my talk. So what is desiccation tolerance? Oops, my slides don't seem to be working. That's interesting. Sorry, guys. I think my slideshow was stalled. Anyway, doing it that way. Desiccation tolerance is the ability to survive, and depending on what literature you're reading, the tissue can survive lost 2.1 grams of water per gram biomass. Normally, that's what you'd read in the seed literature, and we have about 90% of our seeds are desiccation tolerant. Alternatively, it's defined as the loss of up to 95% of total water content. And this is in the field I work in vegetative desiccation tolerance. And we normally refer to this, as you'll see through my lecture, as relative water content. Or if you're a plant physiologist, it's the ability to survive approximately 100 megapascals for a long period of time. And this is actually a very amazing and relatively rare feat. For example, in animals, there's only a couple of examples that can actually dry without dying. And you're seeing some pictures of them here. The tardigrade I want to draw your attention to, because it's the first animal that has been sent out into space and survived. That's because it's desiccation tolerant, and can go through all the huge stresses, extreme freezing, extremes of temperature, etc. And I'm going to get back to that later in my talk. With respect to vegetative desiccation tolerance, it's actually quite rare. This diagram is taken from a review that I wrote with a bunch of people in 2020. And everything you see with the red box around it is a class that has desiccation tolerant plants within them, right from the algae through to the angiosperms. And if you want to look at the quantities, you find that, and this is from Marx et al., that in the mosses, up to 30 to 60% of them are desiccation tolerant. This is a bit of a sliding scale, simply because it is believed that most mosses can be made desiccation tolerant if you prime them properly. The lycophytes, about 20% of them are desiccation tolerant. And of course, these are the organisms that first came onto land. They're small, they dried really quickly. And so most of their protective mechanisms are constitutive. Come evolution of tracheids and spores that are desiccation tolerant in ferns. There's alternative way of tolerating water loss or mitigating the stress of it. And you suddenly find there's a huge loss of vegetative desiccation tolerance, with only 1% of the ferns being able to being desiccation tolerant. Coming to the angiosperms, here we find it drops to even lower, 0.07%. And it evolved, according to Oliver et al., at least 13 times. So there's quite a controversial discussion going on in the literature about whether this is convergent or divergent evolution, and I'll touch on that a bit later. But essentially, all angiosperms are referred to as, or that tolerate desiccation, are referred to as resurrection plants. And you'll have seen that in my abstract. It's a common term, originally not covering the lower orders that are desiccation tolerant, simply because the lower orders are constitutively desiccation tolerant. Resurrection plants actually induce tolerance. They actually lay it down as they're drying. But nevertheless, it's a term that kind of nowadays is used to cover all of these. I work predominantly on angiosperms because all our crops are angiosperms. And I think, as what you know, one of the things that I want to do is make crops more drought tolerant. So I know this is a really redundant slide for most of you in the audience. You know that this is happening. Climate change is for real. But I love to show this particular slide because it was written by Drier in 2011. And most of what he's predicted, and certainly in terms of aridity, is coming true. Now, when he published this, all the areas in red here had been 10 years prior been successfully used for agriculture, no longer possible due to lack of rain. This is his prediction for 2050, which is actually quite disastrous. You guys and myself in our country, a lot of the world, will be too arid to do agriculture as we do it today, as we know it today. And so why is this a problem? And how can we solve this? Unusual. A lot of people get a little bit shocked by this figure, but there are literally, when you think about plant calories that are used in our diet, there are only 120 plant species used for human food. Nine of these supply 75% of global plant-derived intake. So all animals, all insects, or whatever it is that survive on plant, supplied by 9 plants. And of these, 3, wheat, rice, and maize, account for 50% of this energy. So every single one of us are eating a lot of this product, even if though we don't know it. It's probably some in our feed chain, but even in in the products that we eat. Now, all of these plants were bred in the Green Revolution for higher yield, bigger seed size, and as a consequence, there's been a compromise in the ability to tolerate water loss. It was a great strategy right then, you know, that lots of rain, population growth, big seeds, lots of yield, fantastic food. You can store it, times of need for time of plenty. But as I say, it compromised the tolerance of water deficits. So if one looks at wheat, rice, and maize these days, they lose 15 to 30% of their water, they die. Now, the so-called drought-tolerant crops that we say have been produced recently are not tolerant of increased water loss. They either have bigger root systems, deeper root systems to actually mine subterranean water, or they are grown in very short, their breeding cycle has been reduced so that the chances of drought happening is quite slim. But if it's a prolonged drought, which is inevitable, certainly in Africa, and what I was going to say is 95% of Africa's agriculture is rain-fed. So this is really, I mean, I'm an African and I'm really passionate about this. I think you'll notice. But we need to do something different. So what are the solutions? And this is just taken from a review that I wrote with Henk Hilhorst, who's been a wonderful collaborator. He's from Wageningen University of Research. And what can we do to have crops in dry environments? And there are a bunch of suggestions. The first two I'm not involved in, but genome editing of existing crops, if you know what to edit for, this could work. De-domestication of current crops. Is so much GWAS information out there that this is possible, but you are going to drop in yield. Using indigenous or orphan crops. Now, I'm not sure if many in the audience do know what an orphan crop is, and it's essentially a crop that's been grown successfully millennia, same as maize, wheat, and rice. But in areas where it's mostly Africa and South America, where they've not been bred, like in the Green Revolution, and they have not had their drought tolerance compromised. And just a couple of examples. I don't know if you've ever heard of teff. It's actually a grass, so people eat the seed. It's very small, but it's gluten-free, highly nutritious, and has become known as a superfood. If you have been to an Ethiopian restaurant, you will have eaten injera bread, which is made from that teff seed. Legumes. In Africa and South America, India, there are amazing amount of legumes that are extremely drought tolerant, never been bred for, they've never been bred in equivalent ways in the Green Revolution. Here I'm mentioning just three, which are very African. I work on bambara beans. You probably never heard of a marama bean, but it literally grows in the desert. And then finally, one you might have heard of is quinoa. It's from South America, and this is used as a cereal in many other ways. Getting to the nub of my talk. So what about using resurrection plants as models for making more drought-tolerant crops? And for the last 30 years or so, I've been using a whole bunch of resurrection plants. Some of them you can see here. And I point out that they are also heat tolerant. So there are a number of resurrection plants in Africa. I'm very lucky. And all of these, every single one of them can tolerate really high temperatures. Whereas those, and there are not many in North America, I don't think there are any in Germany, are normally dry in order to cope with frost or freezing. So there's no water to have frost damage, freeze damage. Now I work on all these different varieties simply because although we do know that there are similar things that each of these do, and I'll get back to that, there are considerable differences between them. So each of the crops you see, these plants you see here are a model. All the xerophyta species, and I know I don't need to use three, but there are various reasons why I am using three, serve as models for making drought-tolerant cereals, particularly maize or corn. It's a staple food in Africa. It's the second staple in Europe, and also in the USA. So I've put a lot of emphasis on that. Cratrostigma wimsii. In fact, I work on two different cratrostigmas, not the one that Dorothea Bartels has worked on. It's also African, but I've chosen not to work on that because she's put out so much excellent work for those of you who know Dorothea. But this is my model for making drought-tolerant legumes. As I've already said, bambara beans is what I'm doing. Also highly nutritious with incredible antioxidant potentials. Eragrastis nandensis, it's the only resurrection plant that has a crop that's already a close relative, and that's I've already introduced as Eragrastis tef or tef. And in our lab, tef dies below a water content of 50% relative water content. So that's, it's lost, can lose half its water without dying. It's really quite resilient. Myrathamnus fabellifolia is my favorite resurrection plant, and I'm introducing it into the mix today, not because I am using it as a model per se to make, it's a woody resurrection plant. There are only two in the world, and itself and its a sister in Madagascar, Madagascar, yeah. And I'm not intent on making shrubs or fruit trees more drought tolerant. They're already fairly resilient as on their own, but I'm using it as a model for other aspects of making drought tolerant crops, which I'll come to later. Now I'm doing something incredibly and radically different from what anyone is doing in terms of breeding crops. So I've undertaken what we call is a systems biology approach. I'm sure you're aware of it, in which I embrace a whole bunch of techniques in order to comprehensively understand how plants can dry without dying. And only once one has a comprehensive understanding of that can one actually then go ahead and do the biotechnological things. So these are all the various studies I've embraced recently, all what we call the omics, really high-tech stuff, not easily available in Africa, but I've got away with this because I collaborate with amazing people in Europe. And what I'm going to do is actually bring back, and using this approach, bring back my research question. Can this phenomenon of vegetative desiccation tolerance provide answers for food security in a hot to drier future? And of course, I think it can. This is why I'm talking to you. And I'm actually going to tell you 4 stories, 4 phenomena, 4 instances where I think this is possible. In the first instance, can we use what I call the GMO approach, a genetic modified organism, but in fact, using genetics, can we make drought-tolerant crops? Very controversial, but I will approach this. Secondly, using all these amazing omics techniques, you get to be able to answer questions that you just noticed physiologically and perhaps no one else has answered. And one of the things that we have noticed about resurrection plants is that they are able to suppress drought-induced senescence in most of their tissues. Now, when crops go into drought, the first thing they tend to do is senesce their leaves. The nutrients from those leaves go back into the plant and they flower prematurely. Ultimately, it dies. Resurrection plants don't. So, second question I ask is, given this understanding of how they suppress it, can we perhaps implement this in crops under drought? Thirdly, and a review done by one of my PhD students in 2021, and I'll introduce her to you later, where she looked at a meta-analysis of all literature on resurrection plants in the last 2 decades, and found that there's an incredible paucity of studies on roots. About 2 studies, most of us just concentrate on vegetative, on the leaves. So there's a paucity of study on roots, but particularly no studies on microbial association thereof. And we do know that out there is, and I think we call it the unexplored universe of bacteria and fungi that actually totally associate with plants. So knowing this, I ask a question: can we actually perhaps produce a microbial biostimulant to improve plant growth and tolerance to drought? Finally, using Myrathamnus, which in itself can be a food, but also are very useful in many ways. But can we also use this as a model for seeing if we can have crops on the moon and elsewhere? So firstly, the GMO approach. I'm going to talk mostly on Xerophyta schlechteri. In fact, I'm going to talk a lot on this plant during my talk. This story started in 1995 with one of my first ever postdocs. I was new and academic at UCT. And he stayed on as a lecturer for a little while afterwards, but he did a then incredibly important, but now considered trivial experiment called complementation by functional efficiency, in which he discovered 30 so odd genes that we thought were really important and needed to be further analyzed for desiccation tolerance. We subsequently have published some of this. A group of people have gone on to characterize all of those genes, and enter a biotechnologist, Jennifer Thompson, and Suhail Rafadeen, who took over Sagadivan's post when he left. Between them and 23 years later, we finally do have a genetically modified maize plant, which has been field tested at present. That took a long time, lots of money, a lot of controversy doing work like this. But what you're seeing here is the wild type to the left, the plant with a drought-inducible promoter from SARChI. Now, what is important about that is it senses the water content, water deficit, and then switches on the downstream genes, which are two very powerful antioxidants. And you can see that 3 weeks without water, these containing the genes are doing considerably better than those that don't have the genes. Although what I must say is that they do better under drought, but they're pretty much what we're doing with a lot of other plants these days. They do not tolerate more water loss. So that brings me to the question, can we actually bring about this resurrection plant phenotype? I was very convinced in the beginning of my research career that this is possible. I'm less convinced. That doesn't mean to say I don't think it's going to happen. I think it can, but probably beyond my lifetime. But what we are doing is a significant amount of studies. So I mentioned that there was something similar between all of these plants, right? They did similar things. And what we are able to show is that many of the genes that resurrection plants are using in their leaves are actually genes that have been described or thought to be totally associated with desiccation tolerance in seeds. So if you look at this cartoon that I've got here, if we assume, and it's quite simplistic, that all the genes associated with desiccation tolerance are on this chromosome, they need to be turned on during desiccation tolerance for the seeds to survive. We have shown, and not only myself, a bunch of people, that similar genes are used in leaves and roots. Ergo, all crops which produce desiccation tolerant seeds actually have the genetics. They just never switch on these genes in their leaves. They keep them firmly suppressed. Understanding how to switch them on by using a resurrection plant could make that plant a lot more drought or desiccation tolerant. And we are trying, we're looking at a number of regulatory elements and doing CRISPR-Cas at this point in time. Turning to my second question. How do they suppress senescence? And is there a biotech application possible? I'm going to talk about, actually, again, Xerophyta schlechteri, but we've done this on 3 species. I didn't add the dicot image in here, but using all the omics, recently completed on the grass, Eragrostis nandensis. An important thing to notice that both these are monocots, and both of them have a strategy of, monocots tend to do this. They have a strategy of breaking down their photosynthetic apparatus during drying because it's such a damaging and dangerous piece of metabolism, and then having to rebuild them during rehydration. Now, as I said, I'm going to talk mostly on schlechteri, but having looked at these studies and one on Acraterostigma, we can make some common observations. First is that when you, and just to let you know, so the tissues that senesce are the oldest tissues. And in the case of schlechteri, I'll show you it's the tip of the leaf. In the case of nandensis, it is a whole but older leaf. So when they start to dry down, there's absolutely no difference in transcription or proteins made or anything. They actually do initiate a desiccation tolerance program. But at 40%, now this is a critical water content if you're looking at plants. Dorothea Bartels was the first to say that if plant can survive below 40%, it can be classified as desiccation tolerance. If not, it's drought tolerant. And I agree entirely that it's a tipping point, but resurrection plants have got to have prepared sufficiently to get to that tipping point for them to survive. If not, as a drought-tolerant plant, which normally, a crop would normally be at that point almost dead already. So at 40%, you suddenly get the senescent tissue, the older tissue, behaving like a sensitive tissue. The transcription proteins produced, and particularly the metabolism, is evidence of senescent tissue. But this is very different from what's happening in the non-senescent tissue. And finally, senescent tissues never rehydrate, so their job is virtually done by the end of the desiccation curve. Now, looking at Xerophyta schlechteri, and I'm slipping in flowering here. This is a plant that's survived 2 or 3 cycles of dehydration and coming back to life. And you notice that there's these long, they are dead tips to the leaves. That's the oldest part of a monocot leaf. These little purple bands that kind of straddle between the dead part and the alive green part. So a transcriptomic study, followed by a proteomic study by another student, was done by a student called Astrid Rademacher. And what she essentially found was, now this is a plant that's never been dehydrated again. So at fully hydrated, there's only a small evidence of perhaps a bit of a browning on the tip. By 60% relative water content, you get a lot of purpling on the outer side of the leaf, and the leaf starts folding down the blade, as you can see here. But that is a marker for the fact that almost all of that leaf is going to die on rehydration or during desiccation. In the dry state, the whole leaf has gone purple, it's completely folded. But when you rehydrate it, you find that only greens up to just before this purple band, and then the rest has died. So what she did is compared changes in gene expression between tissue that's destined to die, senescent tissue, pre-senescent tissue, she called it, tissue that's going to die next round of dehydration, and then non-senescent tissue, but she took it right from the base near where the meristem is. And I know this is a complex graph, but she looked at changes in gene expression during drying and rehydration, used k-means clustering to cluster data. So in this case, it's similarities in gene expression. She arrived with 12 superclusters. And just to explain this, each of the little gray areas here refers to a gene expression, transcription of genes in the non-senescent, the pre-senescent, and the senescent tissue at the water contents of full turgor, 75, 55, 35 in the air dry state, and then 48 hours in rehydration. What we've got here is that this is all color-coded to help with interpretation of our network analysis. But what you can see essentially colored in green are genes that tend to decline a little bit during early drying, but massively increase during rehydration. Not so much in the senescent tissue, but I'll come to them later. But in each case, you find this nice increase during rehydration. So we dubbed them recovery genes. In pink, genes that actually had an early response to dehydration. In blue, they had a later response to dehydration. In purple, almost similar, but no real changes. And then various shades of yellow and orange, genes that are really different in the late stages of drying and rehydration. And that is, of course, the senescent tissue. So doing network analysis, you find really interesting clustering of genes in recovery, in green, early response, non-specific changes, but really important down here are changes that clustering of genes that are tip-specific, in other words, they're senescent genes. Zooming into this point here, there's one gene that connects the recovery and the senescent groupings. And we've dubbed that the anti-death gene. Now, one of the things about resurrection plants, lovely to work with, but there's absolutely no way of transforming them, knocking out genes or anything. Most of them are polyploid. So we moved to Arabidopsis, which everyone does. It's such a well-characterized model. And this is the work that came out of Henk Hilhorst's lab, one of his master students, where we used a tDNA knockout of this so-called death gene. And we found that the wild type here on the left, by 30 days, pretty normal, but premature flowering in the knockout. After 36 days, you start getting premature senescence happening in these plants. So given that, and we've still got to do the reverse experiments, one would assume that overexpression might prevent flowering and stop senescence. I'm going to take a pause right now and show you a video of the resurrection, the poikiloc, well, sorry, I haven't used that term, monocot resurrection plants coming alive again. Remember, I told you that they break down the thylakoid membranes, their chloroplast membranes, so they take a bit of time to rehydrate. I'm also going to show you the recovery of a dicot, which tends to keep its chlorophyll, so it recovers hugely quicker. The one strategy associated with monocots, and I've been talking about monocots all along, is that they, along with stopping senescence in most of the leaves, they actually stop flowering. And you can guarantee that they will flower afterwards. Have a look at the two. So after 4 hours, the dicot, craterostigma, pretty much fully hydrated, and its flowers resurrect. After 12 hours, 24 hours, you start seeing a lot more green. You can see the senescent tips. But after 3 days, without fail, going on a bit further, you get flowering. And just to say, to a subsistence farmer in Africa, either of these strategies are great because you guaranteed seed after a drought. And that is a really cool thing, because seed is what you trade on. Turning finally to my favorite resurrection plant, Myrothamnus frobilifolia. It's in the dehydrated and hydrated state, and you can see it grows on rocks. And in fact, it excretes organic acids to break down the rocks in order to make soil for its roots. And I'm using this plant, I'm going to answer in part. Number 3, can we develop microbial biostimulants? And 4, use in other purposes other than just food, but also paving a way for food on the moon. Now, this is a study just completed actually by my PhD student, Chandri Tubele. She is interested in ultimately looking at microbes that can be used for biostimulants, but she took a bottom-up approach. And she looked at metagenomic, well, she sequenced them. She did metagenomic sequencing, metatranscriptomics, and metabolomic studies. And I'm going to show you a couple of these. Now, importantly, Chandri's work was all done, all the collection was done in the field. In the resurrection plant literature, most people either start from seed in soil, potting soil, so they completely don't know what's going on underground, and it's not natural. And if one's looking at microbes, of course, you have to be very careful that you don't include any strange ones when you're transporting these things. So, starting first with the metagenomic studies, a little cartoon shows you that when you look at the cross-section of roots, there's what we call bulk soil, and there are a whole lot of microbes in the bulk soil. Rhizosphere soil, which is soil closely associated with the rhizosphere, and there are microbes there. And then endosphere is where microbes actually are invited all the way in, and they work in symbiosis together. And I'm going to get to the metagenomics results, but I also just going to go over a little bit further to show that the girls, and I say the girls, Rose Marks is a postdoc, and I'll introduce you to her a bit later, Chandri, wanted to do an in-field drying and rehydration experiment. And as you can't always predict when weather's going to happen, when they got to the field, the plants were already partially dehydrated. They then allowed them to desiccate and waited, and the trip was it takes a 2-hour plane ride and a 5-hour plant road journey in a 4x4 to get to this field. And they needed to get back. So they made a plan. I'm going to start the video to actually bring... It's not actually working. Anyway, to bring water to the field. If I can get the video to work, what actually is happening is that you find that they actually got lots and lots of water here, and they sprayed the field. It could be possible that I actually need to start talking more about the top grass. So when they got at each of these sample points, they actually isolated roots that went for a separate study, and I'm not going to talk about it today. And what Chandri did was then sift out the microsphere soil, which she then did transcriptomics and metabolomics on. Oh, there's the video going. So here the girls are trying to get the plants that they had labeled and following rehydrated. Life in Africa. That's Rose. So I said I'd introduce you to Rose. She's an avid postdoc. She actually spends most of her time in MSU, Michigan State University, with Bob van Buren, but she does do some work with me at UCT. So getting back to Chandri's metagenomics study, of course, she sequenced in all those three areas. She did alpha and beta diversity, relative abundance, soil, effect of soil factors. And then we had a list of genes, of candidates that occurred in each of these three zones. But we found, and this is bacteria, that were particularly enriched in the bulk soil. You had Coleophaceus and Leptoglymbia. These are known to improve biological conditions of the soil, so helpful to the plant. In the rhizosphere, microbes that make indole acetic acid, it's known, and we show this also in the metabolomic studies, that this helps root growth. And then bacteria that were invited right into the root, into the endosphere, might have been there already, both the two main families produce exopolysaccharides, which is high nutrient for themselves and for the plant. Amongst the fungi in the bulk soil, inhibit pathogens. This is quite a cool thing if you want to making a biostimulant. You don't really want to have other pathogens coming in the field. In the rhizosphere, fungi that produce proteases and lipase enzymes, which again indicates breakdown of lipids and fats for nutrients. And in the endosphere, fungi that are resistant to heavy metals. Again, it might not be fabulous for general biostimulant, but for a farmer who's got very poor soil, this might be a useful candidate. So this has all been published. And just to show you a small amount very quickly, I'm aware of time, amongst the metabolites found in the rhizosphere soil, looking at the amino acids is quite a strong accumulation, and red is dry, yellow is partially dry, green fully rehydrated, and purple partially rehydrated. Lots of amino acids, including proline, which is known to stabilize proteins. During rehydration, partial rehydration, and full rehydration, you see popping, again, a number of amino acids popping up, which could be of great use to the plant. Sugars are well known to be part of desiccation tolerance. Pretty much every resurrection plant accumulates sucrose. In the soil, we did not get sucrose, but we did get trehalose. And this is well known in the animal literature to be a water replacement molecule and a signal. During rehydration, whole bunch of, including sucrose, of really useful metabolites pop up. And then amongst organic acids, and we found that these only occurred in the partially dry state, but are known to be helping nutrient acquisition, particularly with hormones. And if I can get my pointer to work, I can show you there's indole acetic acid, which is known to enhance lateral reproduction. So a lot of pro features about these bugs, but do they actually work as a biostimulant? Chandri has done that work. I can't actually talk about it any further because it is going to be patented. But I am going to talk a little bit about the subject using a top-down approach, using a different resurrection plant, Xerophyta schlechteri, work done by my PhD student, Joel. And what we did here is we just took from the dry soil, we just took mostly bulk soil, but some rhizosphere soil from the field, brought them back to the lab, and we only wanted to inoculate with the soil. We didn't want to use the soil entirely. And so we sterilized coca peat and then added either soil that's been sterilized 3 times in 1-week intervals, or unsterilized soil into pots, grew maize plants, droughted them or kept them well watered, and drought was for 10 days. And then she compared what happened without an inoculum and with inoculum. And essentially, there's a massive increase in plant growth with inoculum, as you can see here, compared to the control. After 10 days of drought, the plant survived a lot better. You can see under control conditions, leaves were in this inoculated were much bigger. And after 10 days of stress, there's some curling in the inoculated leaves, but a whole lot of senescence starting in the non-inoculated leaves. Probably the most exciting is the massive amount of root growth that occurs in inoculated species. So she did a whole lot of biochemical work as well, and what she's shown is that leaf relative water content was better retained. They had there was higher protein content, sugar content, antioxidant content, photosynthetic pigments better contained, less oxidative damage, and an 80% improvement of root growth, 50% in leaf growth, and 100% prevention of senescence at that point, compared to 50% senescence in control leaves. I was going to talk briefly about this slide of Myrothamnus favellifolia to indicate that during drying, there is massive accumulation of a whole bunch of secondary, or what we call specialized metabolites that have a whole bunch of medicinal properties. I'm very aware that I'm running short on time, so I want to move on. So this is essentially Myrothamnus is a medicinal plant, and for centuries, as folklore has it, it's been used as a tea. It is actually on the supermarket shelves in Zimbabwe. And increasingly, it's been used in cosmetics. And one of the biggest culprits is Giorgio Armani and his Rein Crema Nera Extrema. Now, I know this. I used to be consultant for them, and I'm highly impressed with him. I have retired or resigned from that post. But you can see that the data shows, and this is a green extraction, Giorgio Armani are part of L'Oreal, that there is, and this is just for interest, ladies in the audience, that there is a massive increase in differentiation of the cells, particularly the dermal density, just after 35 days of treatment. And so medicinal plant, it's going to be of value. It is of value to humanity. And we are further doing tests in the skin and hair clinic with one of my PhD students, seeing if it can work for wound healing and burns, because this is, again, traditional knowledge that it does. So it's an important plant. If we're going to do anything, it might become a bit of an icon. But I'm concerned that there's been no studies done on sustainable harvesting of this plant. It grows extremely slowly. We know how to germinate it, but we might drive it to extinction if we are going to continue to use it at the rate at which it's being used. So I've started something called, I'm sure you've heard of Save the Rhino campaign. People want to use rhino horn for many medicinal purposes. I'm going to be polite about what one in particular. But I've started the Save the Myro campaign, and I've tried to convince my government that if we could get some money to actually find out how this could be used sustainably, it could make a whole lot of subsistence farmers a lot of money in the future, especially those living in very arid areas. Nevertheless, I have not met with success, so I've taken it one step further, and I'm trying to alert the world to the amazing potential of this plant. So what am I doing? I'm sending Myro to the moon. Very dramatic entrance, but in fact, there's a bit of a backstory. So in 2019, an Israeli company called Space Isle launched it was the first privately owned craft to be launched to the moon, and they attempted a landing, were not successful, and are attempting to do this again in late 2024 and 2025. And they set up a competition for anyone in the world who could show that they have a very valuable project to do. That team, that project, would get a free payload to space. So Lileria 1 was born, and I was invited to join. I'm just a mere biologist, but it's a huge consortium of space engineers and all sorts of people, with the aim of growing a selection of plants on the moon. Now, it sounds grander than what we really are going to do. We just want to see if seeds, and I mentioned earlier, if you go out to the moon, you've got to have a lot of things that you withstand. Seeds can withstand desiccation and extreme temperatures. So can a resurrection plant. So the first question is, can we get them through all of that and see if they will resurrect in space? And then we know something for the future. So why Myro? Well, first of all, it's very photogenic. The grasses just aren't. And the seedlings, unlike many other resurrection plants, are extremely desiccation tolerant. So even at the 2-leaf stage, it can dry down, and within 24 hours, it's back looking exactly like it was before drying. In the desiccated state, it can survive minus 80 degrees for over a month. We haven't tried longer. So it's an ideal candidate to go to the moon. So it's one of the things about our program is that we've got a huge educational outreach. We want to bring in citizen scientists. All of our data is going to be freely available. And through your computer, you will be able to watch the germination and resurrection process while it's happening in real time. This is a mock-up of the device that we're going to send up. It's a lot more sophisticated now, but essentially there's a camera that uses the moon as a backdrop. There's a seedling tray or resurrection plant tray there. It gets put onto the moon. A water dispenser puts water into the seeds, and there's live photography for 72 hours. I, at this point, have finished talking, and I'd just like to acknowledge that I have had amazing financial assistance for my research, all of these listed here. I also need to acknowledge I've tried to do this as I've gone along, but all my students and collaborators who have made contributed to my work. And of course, I'd like to just, I haven't done any money, spent any money on the Humboldt towards research yet. I'm going to, but I just want to acknowledge this. And if I have time at the end, if the questions allow me, I would like to advertise a conference that I'm holding in January next year, in case anyone might be wanting to join us in Africa, see a whole lot of wildlife, but also do some amazing talking and networking about water in plants. Thank you. I'm not going to, well, let me stop sharing for now. I'll start sharing again if I have time to show my advert.
Okay, thank you very much, Jill, for this very, very interesting introduction in the various aspects of your research. And I'm pretty sure that there are questions. So I would like to open the question and answer session. And I see Peter, please start.
Yeah, hello. Yeah, very nice presentation. I have a question to the last part on this experiment where you took rhizosphere soil bacteria, as I understood, to the soil to grow maize and to demonstrate that then the maize shows improved drought tolerance.
Yeah.
I wonder about the mechanism. You showed that the root proliferation of the maize plants is, the roots proliferate, they grow better. Is this maybe this is just speculation. Do you think this is due to hormone effects by the microbes in the soil? And then is the root proliferation causal, or the reason why the plants do better under drought because they have access to more water from the soil?
I think that's exactly it. I think you've hit the nail right on the head. We have shown, and it is known, not just my work, but that a lot of bacteria, some fungi, do produce indole acetic acid, which is a root hormone. And you can see that it allowed just growing in, the plant growing with these bacteria. You really did see a massive root increase. Now, because there's a massive root increase, it's what we're breeding for these days, is better. It can mine better the water that's available and send it up to the leaf, so the leaf can grow bigger, they can bigger yield in seed, and so forth.
Are you going to identify these bacteria? I mean, this would be great, and then have this bacterial strain to like inoculate all kinds of drought.
This is the whole idea. So when I say we make a biostimulant, we are in the process of identifying them. We will make a cocktail of the ones that we think are going to be best to which to prime seed. So you prime them, you dry them back, because all of these are desiccation tolerant. I didn't stress that during the meeting, but everything we're identifying can dry. And then you put your maize out in the field, and we're trying on lots of different crops. I'm just not allowed to say too much right now, because I think you can believe it's patentable. and I've got people really interested in investing. So, but I do, I've got to the point that people are so interested in investing because it's true. We are seeing, we've got to go and test it in the field. We've got to test whether these microbes are going to fight with other microbes in the field. There's a lot of testing that needs to be done. But I really strongly believe that this could be a solution, certainly to farmers in Africa who don't have sophisticated, or they can't, that they don't have fertilizers and things like that.
Okay, thank you.
Thank you. Further questions? I have actually one, Jill, because I never thought about it before in all our discussions. You mentioned at the very beginning that there is a number of plants who do not have desiccation-tolerant seeds.
Mm.
So when you look at these plants, are they actually missing out these whole genes, a set of genes that are turned on in the desiccation-tolerant seeds, and therefore will never be able to be made into desiccation-tolerant plants, or are they just not switched on? And can we improve these maybe for our purposes by even making them seed desiccation tolerant?
My grin. It's big. Yes, it's a stunning question. It is something we are tackling right now. And I say we, that would be Henk Helhorst and myself, who I introduced during my talk. So the seeds that are not desiccation tolerant, we call them recalcitrant seeds. And during development, when a normal seed becomes desiccation tolerant, it never expresses those genes. They actually just go straight into germination. And the rate at which they go into germination does vary between the species. But our recent results have shown that the regulating factors are present. They're just never expressed. So ABI3, for example, is a well-known regulator of seed desiccation tolerance. It's present in a recalcitrant seed called Casanospermum. It's just not expressed. And so coffee, amongst the recalcitrant seeds, there's coffee, some citrus species, avocado, lychee, rubber, a whole bunch of economically important crops. And we have just, in fact, I can actually say it now because we've raised money. We have funding to actually try and make coffee seeds desiccation tolerant. And why that's important is, I don't know people who love coffee, but in Brazil and in Africa, where a lot of coffee is grown, the droughts have killed off the trees. And so you can't store the seed, you can't germinate, and it's a problem. And I'm absolutely excited. So the interesting thing about this is I did much of this work in my PhD. And it's come back to bite me now.
Okay, great. So in the moment, I don't see a raised hand, so I will take a chance to have another question that occurred to me when I saw the beginning of your talk, when you also box the algae with regard to desiccation tolerance. So I have worked a little bit on algae during my PhD, actually. And I can imagine certain stages of their life, when they produce kind of spores to be desiccation tolerant. But if I think about a normal algae, I have a little bit of a problem to see, how desiccation tolerance can they get?
Yes, big question, good question. And again, I'm not totally familiar with the number of algae that are desiccation tolerant, but people who work on areas where there's a huge intertidal range. So in some species, there are going to be algae or small associates and rocks that are exposed to desiccation for long periods of time during a day or even 2 days. And those have been proposed to be desiccation tolerant.
Mm-hmm. Okay.
Peter?
Okay, I have another question, maybe related to Ute's last question. If you look into the animal field or bacterial field, I guess you will also find desiccation tolerant species. Did anybody ever look into the genes that are relevant and compare those with the genes important to provide desiccation tolerance in plants? Is there any overlap between bacteria and plants and animals and plants?
There is a small overlap. So desiccation tolerance per se can be achieved following certain criteria, but the chemicals that make those criteria differ. So in tardigrades, bacteria, and things like that, they accumulate a lot of sugar, but they never accumulate sucrose. They accumulate only trehalose. They do make, even recently, even in some of the lower organisms, mosses, for example, they find that there is ABA regulation, something that higher plants do with ease. So to some extent, the primitive or lower order species do have things in common with plants, yes.
Anything known about animals? Is there any desiccation tolerant animal?
Nothing bigger than a tardigrade or a nematode.
Mm-hmm. But and what do they do?
They accumulate shitloads of trehalose and some Lear proteins. I'm not completely up on the literature, but they protect like hell, and it's almost constitutive, so they have a lot of it present. As the drought hits, they'll increase it a lot and then repair afterwards.
So how conserved are these Lear proteins? That's actually quite interesting in evolution.
The evolution in the angiosperms has been huge, and it's such a difficult field to understand. But there's in the lower plant species, Selaginella, for example, there have been papers to say there's a Lear-like protein. And I think they're involved in gene regulation, to be honest, amongst other things. But yeah. Mm-hmm. Time is nearly up, and if there are no further questions, may I advertise quickly?
Sure, go ahead.
Right. So what I'm wanting to advertise here is that in January, the 15th to the 19th next year, I'm holding a workshop. I call it a workshop, not a conference, because I like that to give times for people to interact in social conditions, talk about work. And it's got a broad title, Desiccation Sensitivity and Tolerance Across Life Forms. So if you work on drought tolerance, if you work on desiccation tolerance, if you work on sensitive seeds, if you work on animals, you're invited. We're wanting to try and explore this the whole mechanisms of improving drought in every way we can. Have a quick glance at our keynote speakers. You'll see Utter is a keynote speaker. It's going to be held in quite an elite, a 4-star place. It's called Euphoria Golf and Lifestyle Estate. It's got one of the premier golf ranges. So if you're a golfer, bring your clubs. It's apparently it's a signature course. There are a lot of other things. You can see animals, and the cost, very briefly, this includes, that might seem a huge amount because you've still got to pay your airfare, but this includes everything you need. So the moment you land, you're going to be transported, you're going to be taken to the conference, and you will be fed. And if you want to do extras like a game drive, that might cost a little bit extra. So if there are any students, we are considering reduced rates for students. If there's anyone wanting to come, please feel free. We've got space, and we'd love for you to know. This advert, I'll leave with Utter, or if it's still being recorded, you can take the details. That's our email address. This is our website. And when you get to the website, you just have to press register, and things can happen for you there. Thank you for allowing me to do that.
Yeah, thanks, Jill, and thanks for this very, very interesting presentation that you gave to us. And I had another epiphany just in the moment, because when you said there is wildlife there, I realized when you say wildlife, I'm thinking about animals. And yes. I think that's actually what happens to many, many people. And I think one of the important things that we should start working on, that when people hear wildlife, they should also think about wild plants that are interesting and worthwhile to see wherever you go.
I agree. Not on the place, but a little while further, there are lots of resurrection plants, so you can actually see them in real life, IRL. Thank you, guys.
Great. Thank you. With this, I would like to hand back to Ute.
Yeah, thank you very much. Thanks very much for talking. Thanks very much for the discussion. I think we're here to close the session, and I look forward to see the audience on the next lecture in the innovation series of TRAS 6. Thanks so much. Take care and goodbye.